thank you for the organizing to give me the opportunity to bring some reflections about water in a place in Argentina that is Santiago del Estero, that water is very scarce. It's one of the driest places in our country. But in these places, the policy is now to create some research institutions, and that's why from 5 years ago we moved from Buenos Aires, the capital, to Santiago del Estero to start to some research about biophysics, and specifically biophysics in lipid membranes. That is the topic in which I want to talk about. Specifically, I want to address your attention to something that in this conference has not been treated in detail, that is the presence of membranes, specifically lipid membranes, in the cell structure, and to correlate the presence of lipids with the formation of a specialized water structure in terms of what I have named Aquanomics and Waterons, and some definitions that I will discuss as the last in this talk. As you may know, and the previous talk was a very nice introduction to these ideas, is that the cell is a self-assembly, self-organized, and self-regulated systems that operates as an open system with the exchange of matter energy and the principles of the minimum energy cost, and also with an entropy cost in terms of organizing the interior at the expense of the disorder of the surrounding. And the function is the cell of the cell is that it is operative. That means it grows and reproduces always in the presence of water. There are lots of experiments in which the cells has been trying to preserve in the absence of water using such as sugars and even some amino acids, but that only preserves the structure. If you want the organism to respond to the media, to grow and to reproduce, you need liquid water. As I discussed before in the previous talk, the cell now can be considered as a crowded system in which mostly are interfaces between the protein and water, and interfaces of the membrane and protein, and DNA and protein. And this self-organization implies organization of functional and structure of water, and membrane interfaces that are specific for membrane response. In terms of water environments, and taking into account the cell dimensions, I mentioned I have to recall that membrane interfaces is a place in which water organized in a 1 nanometer thickness, in which around 20 water molecules organize around the lipid head groups, and that water has specific properties like restricted environments, in which we can find different properties in terms of thermodynamics and structure in comparison, in comparing water with bulk properties. The main driving force that I will try to expose here is the surface tension of water. You know that in water there are some molecules that are not able to complete the hydrogen bonding and lose the tetrahedral array in the bulk, and then they form some special of residual forces that can be reduced by the stabilization, for instance, of a lipid monolayer on the surface. And in that, when we increase the lipids in the monolayer, we change the surface excess, and we have that the surface pressure reduces along the increase of the area per molecule. That is, when we have larger area per molecules, we have a very low difference between the surface tension of pure water and the tension with the lipids. This area is in which we will find differential responses of the membrane. So the point is that we want to explore the thermodynamic properties in terms of surface free energy, in terms of changes in surface tension, to explore the propensity of the biomembranes to respond to molecular events, such as the interaction with amino acids or peptides that could be signals for the biological performance. And in this topic, we will also analyze the water nanodomains as molecular antenna that may be responsible of this response. The lipid monolayer can act as an open system, and in which we can compress the monolayer and extrude water from N, and expand the monolayer and permit water in between the lipids. This has been recently, a paper recently in which for the first time we have applied the principles of the thermodynamics of irreversible processes in order to understand the behavior of the lipid monolayer as an open system in terms that it can exchange water at different stages. And here it is represented that the flux of water is related with the interaction of water with lipids, and the lipids with lipids. All of these permit to reproduce very well the very well-known surface pressure versus area per lipid curves, in which we can see the states of the thermodynamic states of the lipids along the at a constant temperature along the pressure and the area. Sometimes this has been described as a gas on the surface, but the thing is that we have to consider that this gas is not a real gas, but it is the compromise is that the lipids are interacted with water. So when we have a specific state of in this curve of the surface pressure and an area per lipid, we can add to the surface of the monolayer some biological effectors, for instance, an amino acid, a peptide, any molecule that is biological relevant, and we see that the surface pressure still grows, and means that it has some kind of response. And this response is variable with the surface pressure. When the surface pressure, that means the compactation of the lipids on the surface, is larger, we have low response. So we have an intersection in which this depends of the hydration of lipids, that depending of the lipid species. For instance, fosfatidylethanolamine has 2 to 6 water lipids, and the fosfatidylcholine has 12 to 20 lipids. But if at this stage we compress the monolayer to leave only hydration water, we have no response. But if you relax the membrane and we permit more water beyond the hydration gel, we have the response. This same data is explained in this, in which the membrane response is due to the increase in the area per lipid, that is in terms of the increase of surface tension. The different lipids head groups respond in similar, while the change is maintained. You see, these two slopes are similar, and the increase in area means that there is increase of water beyond the hydration gel that is represented, for instance, in this figure, that is water around the principal groups of the phospholipids. So when we fix the head group, for instance, we take only one kind of lipid, but we change the acyl chain, we see that the slope varies accordingly. So the response of the membrane is when we have confined water in between the acyl chains, that is the water that allows the membrane to respond into the to the signals or to the biological effectors. So in this part of the talk, we have the situation that we have expansion of the monolayer, we have a water in, we have increase of confined water, and we have a responsive membrane. When we have compression, we have water out. We have only aeration water, we have a critical packing, and no response. So in the first conclusions is that we have two kinds of water in the lipid membrane, the hydration water and the confined water, and the domains are confined water. The domains of confined water appears beyond the hard core of hydration gel, making the membrane responsive, and this confined water determines the surface free energy of the lipid interface. And these confined waters may be modified by the lateral pressure. Let's talk about now the structure of this type of water. We can use infrared spectroscopy to measure the kind of water and the amount of water with the broad band of water here, and we can also see what happened in the carbonyls and what happened in the acyl change. And we can do that in terms of temperature. You know that the lipids have a phase transition in which we have a packet state in which the lipids touch each other, the CH2 overlaps one, and that's why the frequency of vibration and the wavelength is low, and when the temperature increases, the movement of the change allows the change to separate, and we have a higher frequencies. This is what we find here. Populations of methylene groups are connected populations, and we have here isolated populations, and we can, in principle, think that these isolated populations of the CH2 is because there is water in between. So to test that hypothesis, what we do is to study the infrared spectra in terms of the decrease of water activity, and during time we see that as long as the water activity decreases, the CH2 frequency also decreases. So we are going from connected populations in which the acyl chains are packed to isolated populations in which they are separated water molecules. So the confined water that I mentioned before, that was the responsible to the membrane response, may occupy regions between the isolated CH2 populations. So now, which is the aspect of water that we have in these two states? In the compacted monolayer, in which there is contact between the CH2 population, we come this kind of water bands with these populations found by the convolution. And when we have water that had penetrated into the CH2, making this population of CH2 isolated, we have a completely different pattern of water. So during a membrane, a non-responsive response, we have this type of water, and in a responsive membrane, we have this type of water. So putting the same data in another way, what we have is when we have an increase of isolated methylene population, there is some far apart, we have an increase in the chemical potential of the confined water. And this increase in the chemical potential is the driving force in order to the peptides or amino acid or any significant molecule may affect the membrane, as you may see in this picture. So when we go from a compacted state to an expanded state in the membrane, we increase the amount of confined water beyond the hydrated groups, and we see that most of the groups are affected in terms of the increase of the number of water molecules. But I will detail somehow what is happening along the carbonyl groups. Why the carbonyl groups? Because the carbonyl groups are specifically situated in the interface between the water region and the hydrocarbon region. So, when we compare the response of the membrane of a membrane that has carbonyls and with the membrane that has no carbonyls, we see that there is a change in the slope. That means that the confined water that you can find in a membrane with CO groups is different in which that CO groups are absent. So the CO group is some kind of modulator of the confined water. You can see here a detail of the carbonyls, and these two carbonyls in the SN1 chain and in the SN2 chain has in turn two kinds of population. One is an hydrated population, that is this one, and a non-hydrated population, that is this one in terms of that appears at frequencies, higher frequencies. In terms, to be strict, in reality, we should talk about bound species or unbound species, because this reflects that the carbonyls can interact with other groups besides water. If we have a fully hydrated membrane, we have a 55% of the populations of the carbonyl that is bound, so I mean interacting with the surrounding, a 45% that is unbound, that is without interaction. And when we dehydrate the membrane or lower the water activity to 0.1, that is around 10% relative humidity, we see that there is an inversion, and we have a 40% of bound population and 56% of unbound population. But we do the same experiments in the presence of amino acids such as glycine and betaine, that are amino acids that usually appear in the preservation of plants that are able to survive in dry media or at a low tension water. We see that we obtain the same pattern in the carbonyls than if the membrane is fully hydrated. So, what this data allow us to try to establish a new model of lipid membrane based on the water properties. We have a level hydration water that is expected to be displaced from binding properties, that is, that the amino acids are able to displace that kind of water that we have named as confined water, and these so-called unfavorable water molecules are groups of amino acid, is a principal driving force for binding and the membrane response. The model implies that the surface pressure that we took as the measure of the membrane response is directly related with the water activity. And of course, the surface tension, that was the property that we explored of water, is related with the difference in the chemical potential of water in the bulk and the chemical potential of water in the surface. And in this model, the interface behaves as a bidimensional solution of hydrated polar groups and confined water. And confined water determines the chemical potential of that water that drives the response of the membrane. So, if we take a little bit of thermodynamics and we make the structural link, we can express the activity in terms of virial activity coefficients, and this activity coefficient can be split in a virial, in which we can express all the possibilities that has a water molecule to interact in terms of the hydrogen bonding. And in fact, if we overpose those populations that we found in the compacted state, that is pure hydration water, and the population of water that we may find in the expanded stage of the membrane, that is the addition of confined water, we can see that there are a shift of populations between water molecules that have one non-hydrogen bonds, 1, 2, 3, and 4 hydrogen bonds. So what is the perspective in this? When we see that there are molecules that may have different numbers of hydrogen bonds, we can think that these water molecules can be interacting with other water molecules or with the different groups of the membrane, for instance, the carbonyls and the phosphates. So if by molecular dynamics we could publish this paper in which we have the distribution, and you can see that there are molecules that have hydrogen bonds with water, with carbonyl and phosphates, and we see that this is a population with one water molecule. This is 2 hydrogen bonds with water. Here we have with water, but appears also species that are bounded also to the carbonyl groups and to the phosphate groups like here. So this, we have a multiplicity of states of water depending not only of the number of hydrogen bonds that it may form, that also with the quality of the hydrogen bond that it may form with different groups of the membrane. And that's part of the curve. I don't know how I am in time. The last slides, I will talk about the prospect of this data. Nature has provided a great variety of diversity of lipids. Sometimes the question is why there are so many lipid species. But the problem is, well, the question is that lipids organize water, and is the other way in contraposition to the other prospect in which it is known that lipids organize because of water, because they tend to form, for instance, micelles or by layers spontaneously, something that is related to the origin of life because it forms the compartmentalization in which macromolecules can find a special medium. But in addition to the self-organization of lipids, the lipids itself organize water. And these lipid head groups protrude from the membrane interface where the carbonyls are to the water phase, promoting different water organization according to the type of groups. And the head group of hydration is determined by the cellular species. Remember that this hydration gel has this hydration gel has no effect on the membrane response, but in the change of structure, which is linking to the growing conditions, diet, temperature, hydric stress, oxidative stress, determine the confined water through which the membrane gains the responsiveness to biological effectors. So the conclusion is that we can find in the membrane confined water that is related to the change dynamics and is the backbone in which the hydrogen bond dynamic propensity is necessary to membrane response. It is relevant to, it's a relevant tool for the membrane response to different amino acid motifs. The confined water may be the common intermediary in the interaction of different proteins, peptide amino acids with the lipid membranes, and from this confined water, we can find different molecular organizations named Waterons that exhibit an enhanced propensity to be displaced upon the amino acid association, resulting in stability gain. The dehydration propensity of these Waterons is specific of some amino acid motif. And this work implied a water removal is connected with the binding carbon hydration properties by means of water density fluctuations. So this is the conclusion, and we have also we have edited a book last year about the membrane hydration to those that are interested in the field. I have an exemplar here that you are free to browse. And this was done in the Laboratory of Biointerfaces and Biomimetic Systems with, Maria de Los Angeles made the main work with infrared, Axel Holtman, part of the work in monolayers, and so on, all the stuff in Santiago del Estero, Argentina. Thank you for your attention.
Okay, thank you very much for very interesting presentation and for sharing this. Please.
Yeah, that was very useful for a biologist like me, Anibal, over here.
Oh, right.
Have you done these experiments where you go from the expanded phase to the condensed lipid phase with more than one species of lipid?
Yes.
You have done that because, so my question is, do you find when the domains form, for example, of a lipid like phosphatidylethanolamine and another lipid like phosphatidylglycerol, assuming that they form into separate domains, you would then have a line energy associated with the interface between those two domains.
Yeah.
Is that interface associated with confined water, and does it have a particular chemical potential, and does it act as a signaling mechanism for peptides and proteins to go there?
Yes. Yeah, it's a very good question. Of course, I didn't mention because of time. In cells, we have a mixture of lipids. So we have several, to say the least, we have 3 kinds of lipids, usually cholesterol, for instance. And we can see that the response of the membrane behaves in some way intermediate between the mixtures, and even we have that the slope of the curve that I show you in the here. If you add cholesterol here, for instance, you can change the slope. That means that the packing of the lipid chains is changed by the presence of cholesterol and makes the response of the membrane differently. And if you have different kind of lipids, sometimes you don't have straight lines. You have some kind of in some stages predominates one slope of one lipid, and in other stages predominates the slopes of another lipid. But the question that you raise is very interesting because in membranologists always study the coexistence of lipid domains, but disregard the fact that between the lipid domains there are water domains that are imposed by precisely the properties of the interface between the lipid domains. And that is one of the points that we want to stress, the presence of lipid domain that can be also found in a pure lipid, but is more relevant in terms of mixtures.
Okay, thanks.
So, as you know, in the real cells, the inside of the cell is a cytoskeleton, and outside is often glycocalyx. So it's not a pure, effectively lipid bilayer. How would the presence of those two impact the conclusions that you've drawn?
The point is that the bilayer itself is very important in order to preserve it's a dielectric slab, and And the molecular response, of course, is related also with the presence of some kind of proteins that are in the bilayer itself. But the proteins also alter the packing properties of this. We have done also that kind of experiments in which the insertion of a protein, the elastic and the surface tension changes. And in regard to the exterior of the components in the bilayer, they are not a permeability barrier for that kind of substrates. They can go through without restriction. The permeability barrier for an ion, for instance, or for some kind of elements that the cell needs, goes directly into the membrane. And in the membrane, they have the specific response. Some kinds are related with channels or transported, etc. But in fact, they are correlated with the lipid behavior.
Wouldn't you expect, for example, that the glycocalyx outside, being full of sugars, would actually organize the water around it?
It is possible. The fact is that when that kind of cells are preserved with sugar, for instance, trehalose, you find that the trehalose interact with the lipid bilayer. And that is the reason in which the structure of the bilayer is preserved and the cell is preserved. So the cell cannot survive if the lipid membrane is disrupted. Even, for instance, in many experiments that we have done with antimicrobial peptides, the antimicrobial peptides insert precisely into the lipid bilayer and form a pore. Okay, pore is a way to explain nothing, but I mean that that kind of peptides interact with the lipid membrane and probably making some kind of disruptions in terms of what I am talking about, the presence of different kinds of water.
I like the idea of Waterons. I think that will help organize our thinking. I have one question. As you know, Prigogine divides all the structures in the universe into two classes, right? Equilibrium structures and dissipative structures. Do you have any evidence that Waterons have two classes, equilibrium Waterons and dissipative Waterons?
What kind of water you say that? I think.
You see, Prigogine divides all the structures into two classes, equilibrium structures like tables, DNA sequences, and so on, and dissipative structures like the flame of a candle or action potentials. So I was wondering if Waterons can be divided into two classes. The one that you described seems to belong to equilibrium structures. Is there any possibility for Waterons existing as a dissipative structures, meaning that they exist only if there's an input of free energy? Otherwise, they disappear.
I think the water structure that we are dealing with is labile structure, I mean unstable structures. Because, for instance, in the behavior of the carbonyl, why we choose the carbonyls? Because the carbonyls have two kinds of two contributions. One of the S1 chain and the other of the N2 chain. But in those chains, we have an unbound population and a bound population and unbound in both. So that was a very interesting experiment. So that means that the carbonyls in each chain is fluctuating between going into the membrane or going out, going in and going out. And that we have previous results that were published in Langmuir some time ago, that if you, for instance, you change the topology of the membrane, for instance, in terms of make a curvature or putting lipids with different arrays in the membrane, you have a different pattern of carbonyls. So because of the fluctuation, we think that part of the water is some kind of transient distribution that is between that is the holy hydration, that is a hard core of water around the phosphates, for instance, that you cannot disrupt, and water that is between the lipid change, that is water that can be near an hydrophobic surface. In carbonates, the water have two possibilities: to see a hydrophobic surface or hydrophobic surface. And this is fluctuating and dependent of the environmental conditions. I don't know if I answered your question.